This conference will now be recorded.
Hello students, now we are learning about anatomy of flowering plants, the sixth chapter. So anatomy means here in this picture itself you can see the internal arrangement of all the cells, how these cells are arranged, how tissues are formed, how they are working. About all these things we are learning. Okay, so here according to NEET syllabus, anatomy and function of tissue system in dicots and monocots. That's what we are learning completely about the anatomy and functions of tissue system in dicot and monocot plants. Okay, so already we have learned about the tissue system in plants. So all the tissues of a plant which is performing some or which is doing some general functions, they are forming a tissue system. Okay, so here based on the position or based on that respective of their position we can say. Okay, so not th- all the tissues are joining together and they are performing a function. Okay, it's not based on where they are situated, and also they are taking over a particular function and they're performing to do the activities, they're helping for the activities of a plant. Okay, so according to those activities or their performance, or by considering their uh, work, that important tissue systems are of different types. Which are they? Epidermal tissue system. In the previous class we have learned about a epidermal tissue system, dermal tissue system. It's otherwise called as dermal tissue system. So here you can see epidermal tissue system. And here in the epidermal tissue system, it's clear that which and all part you have learned about the cuticle. This and all coming on the outer side, right? Cuticle, epidermis, then the epidermal outgrowths. You can see some outgrowths like this hairs will form sometimes. So that and all epidermal outgrowths. Then some prickles which is helping for the protection, and also such functions are also there. Then stomata, here you can see all those are coming in the epidermal tissue system. Okay, next we know that the ground tissue system, right? Here in the ground tissue system, or this is a fundamental tissue system, and that forms the main body of plants. The fundamental or the ground tissue system that forms the body of plants. And in monocots, scattered vascular bundles you can see. Okay, here we'll differentiate the monocot stem, dicot stem, the characteristic features of the monocot and dicot, root, stem, leaves, everything we'll learn now. So here In monocot stem, ground tissue system, that is a mass of parenchymatous tissue, and here vascular bundles, you can see scattered vascular bundles. Okay, and in monocot, we can say the ground tissue system is known differentiated into cortex, endodermis, pericycle, and pith. In which one? In monocot. Okay, ground tissue that is not differentiated, not differentiated ground tissue system you can see in monocots. Okay, but in dicot, ground tissue is differentiated in two main zones like cortex, pericycle, pith, and all. Here you can see this is the pith region, and cortex region will be there. Then here this is the cortex region, and pericycle also will be there. Here you can see the pericycle. So this is cortex, then pericycle, and pith region will be there. In which one? In dicot. In dicot it is differentiated, and in monocot ground tissue system is not differentiated into cortex, pericycle, and pith. Okay, so let's see what is this cortex. Cortex means that is the main zone which is lying between the epidermis. Here you can see where is the cortex here. We have marked cortex here. So here it is between the epidermis and the pericycle. Next comes the pericycle, right? So we can mark like epidermis and pericycle. That comes the cortex, and here the cortex portion itself is divided into several layers, right? Here you can see that between epidermis and pericycle, which portion comes? Cortex portion comes, and that cortex portion between epidermis and pericycle, which portion? Cortex portion. The cortex portion is again divided into several layers like hypodermis. You can see hypodermis just below the epidermis. You can see hypodermis. Then this hypodermis, that is made up of collenchyma. Okay, so cortex region, it is again divided into hypodermis, which is collenchymatous hypodermis, and in dicot stem. Okay, and in monocot stem it will be sclerenchymatous. Okay, and this hypodermis region, that is providing strength, and this hypodermis region is absent in roots. See here I have said several points, each one is a MCQ. Okay, so cortex is the zone between epidermis and pericycle. Okay, this is a pericycle region and this is the epidermis region. So in between the portion comes, that is called as what? That is the this is the cortex region, and in cortex region you can see several layers. Here you can see the hypodermis. This hypodermis is collenchymatous in dicot stem and sclerenchymatous in monocot stem. So this provides the mechanical strength, and this hypodermis separation is absent in which one? In roots. Okay, hypodermis is absent in roots. Okay, next about the general cortex. See, cortex, this general cortex that is having parenchymatous cells. Here you can see the parenchymatous cells in the cortex, right? And main function of this cortex. Cortex is made up of parenchymatous cells, and main function is what? Storage. Okay, these 2 points you must learn. Cortex is parenchymatous, and that is made main function of cortex is storage of food. Then endodermis. Next comes the endodermis layer. Here you can see just above the pericycle, you can see endodermis. Endodermis is made up of somewhat barrel shaped cells. Okay, endodermis, that is made up of barrel shaped cells, and these cells are also parenchymatous cells, parenchyma, and it is almost having barrel shaped parenchymatous cells. Okay, and in root endodermis, or the root endodermis cells, they have Casparian strips. Okay, Casparian strips of suberin. In root endodermis, they have Casparian strips of suberin. Then in the case of leaves, what we can say, this and all generally we are saying about each region. Okay, as we are going to differentiate monocot leaf and dicot leaf, then a monocot stem and dicot stem, then monocot root and dicot root. We'll learn the features of each. Okay, then the leaves in this ground tissue is having thin walled chloroplast containing cells. In which one? In roots, because in leaves need chloroplast. Leaves are the sites of photosynthesis, so their cells will be having chloroplast content in that. So leaves, they in their ground tissue, in the ground tissue of leaves, you can see thin walled chloroplast containing cells, which are called as mesophyll cells. Mesophyll cells of leaves that will be having chloroplast. Clear? Then now we have told about the which region? See, we have said about cortex, in that hypodermis, general cortex, endodermis, everything we have said, right? So this is the cortex region. In that hypodermis we said, then the general cortex, then the endodermis. This portion, cortex portion, we have done with that, right? Next comes the pericycle. Okay, here you can see the pericycle region. This is a pericycle. So let's see pericycle. This pericycle that is between the endodermis. See, again we are saying the region between the endodermis and the vascular bundle. Here you can see this is the vascular bundle. So between the endodermis and vascular bundle. Which one? Pericycle region. And you can see here that is parenchymatous in roots and sclerenchymatous in the stem. This is a stem figure. This is a figure of stem. Here you can see this is sclerenchymatous, somewhat thickened cells you can see. So in stem it is sclerenchymatous and in root it is parenchymatous. Okay. And lateral roots that will originate from where? Lateral roots originates from pericycle. Okay, origination of lateral roots, that is from the pericycle region. Next comes the pith or the medulla. See, up to this portion over between the endodermis and the vascular bundle. Next region is the pith or medulla. So that region, that means that is a central portion in dicot stem and monocot root. Okay, generally we are marking the regions. Okay, so here central portion in the dicot stem, that portion also will call what? Pith or medulla. Then monocot root, in that also it is a central portion. So generally it is made up of parenchyma cells. It's parenchymatous. The medulla or the pith region is parenchymatous, and generally we can say that. And in dicot stem, pith cells between the vascular bundle, that becomes radially elongated. In which one? In dicot stem. Okay, and that is called as primary medullary rays or pith rays. Here you can see the primary medullary rays or pith rays. Primary medullary rays. So what is primary medullary ray or pith ray? Here, the central portion in dicot stem and monocot root that we are generally calling as medulla. Okay, so it is made up of parenchymatous cells, and in the case of dicot stem, this pith cells or this medulla cells between the vascular bundle, see between the vascular bundle, the medulla cells or the pith cells that will become radially elongated, and those are called as primary medullary rays or pith rays. Okay, so that helps in lateral translocation. Lateral translocation. Along with that, you should learn the function of each portion also. Okay, so this is what about ground tissue system we need to say: cortex, pericycle, and pith. These are the regions: cortex, pericycle, and pith. While explaining about cortex, you need to mention hypodermis, general cortex, endodermis, then pericycle region, then the pith or medulla region. So here main point is in the case of monocot and dicot, and in stem, leaf, root, in all these you have to learn that is made, each region is made up of which type of cells. Okay, so that's about the ground tissue system. Next is about vascular tissue system. Okay, so tissue system in plants, that is epidermal tissue system, ground tissue system, and the third one is vascular tissue system or fascicular tissue system. Vascular tissue system is otherwise called as fascicular tissue system. Vascular tissue system. See, here actually the central portion, that central cylinder of the shoot or the root, which is surrounded by cortex, that is called as stele. That portion is called as stele. Okay, and here number of vascular bundles which is formed inside the stele, that will vary. Vascular bundles inside the stele will vary. So here that may be of different numbers you can see, and this bundle which is formed inside the stele, that forms the vascular tissue system. And major parts, which are all the major parts of vascular tissue system: xylem, phloem, and cambium, that forms the major part of vascular bundle: xylem, phloem, and cambium. This is the major part of vascular bundle. Here cambium, which is the lateral meristem, which is giving rise to secondary xylem and phloem. Okay, what is cambium? Cambium is the lateral meristem, and this lateral meristem will give rise to the secondary xylem and phloem, and that occurs in the form of a thin strip, that will be in the form of a thin strip, and that consists of fusiform and ray initials as cells. Fusiform cells and ray initials. Okay, so these components, whatever we have said, the xylem, phloem, and all, they have arranged in different ways, and several types of vascular bundles you can see. So we need to learn about different types of vascular bundles. Which are they? Radial, then conjoint, then concentric. All these are different types of vascular bundles. Radial vascular bundles are there. Radial vascular bundle. So in the radial vascular bundle, mainly this is found in roots. Okay, mainly the radial vascular bundles are found in roots, and there you can see separate bands of phloem and xylem. Okay, xylem and phloem bands separately. You can see, here you can see the xylem is separate and phloem is separate, right? So like that separate xylem and phloem bands you can see in which one? In the radial vascular bundles. Next comes the conjoined. See, vascular bundle, conjoined vascular bundle you can see in the stem and leaves. Stem and leaves. Okay, in this both xylem and phloem that is situated at the same radius. Xylem and phloem in same radius you can see. Okay, so both xylem and phloem both are situated at the same radius and they are produced by cell division in vascular cambium. So from vascular cambium it is forming vascular cambium cell division. Due to that, this xylem and phloem, which is present at the same radius of the conjoined vascular bundles, are forming. And conjoined vascular bundles, they are further divided into two types. There are two types of conjoined vascular bundles. They are collateral and bicollateral vascular bundles. Collateral and bicollateral. Okay, collateral vascular bundles and bicollateral vascular bundles. Two types are present in which one? In conjoined vascular bundles. Next, the concentric vascular bundles. Concentric vascular bundles. So in concentric vascular bundles, xylem surrounds the phloem completely, or the phloem surrounds the xylem completely. In which one? In concentric vascular bundle. Here you can see the concentric vascular bundles. Either xylem surrounds the phloem or phloem surrounds the xylem. Okay, here you can see the phloem that is inside and xylem is outside, the red colored one. And in the diagram H, here exactly opposite you can see xylem is inside and phloem that is outside, which is surrounding. So here they are divided, this concentric vascular bundles, they are divided into two types, amphivasal and amphicribal. Amphicribal. Okay, so here concentric means either xylem surrounds the phloem completely or phloem surrounds the xylem completely. So these are divided into two types, that is amphivasal and amphicribal vascular bundles. Okay. And here comes the anatomy of dicot and monocot plant. Okay, dicot and monocot plant, we are going to learn anatomy of dicot and monocot plants. So the primary anatomical structures, which are the primary anatomical structures which we are going to learn, that is root, stem and leaves of dicot and monocot plants. That how we will study, we'll take a transverse section, and by examining that transverse section of these structures we can learn the complete details. Okay, so there will be some common features which is existing features present in the epidermis, and that will be found in the root, stem and leaves. But to know more about this structural differences in their anatomy, um, we are going to learn each one separately, like monocot stem, dicot stem, what and all the differences. Here itself you can do a comparative study. Then the monocot root, dicot root, monocot leaf, dicot leaf, like that we are going to learn. So first, let's start with the dicot and monocot stem. So we know that stem means that is the aerial part of plant, right? Which is the aerial part of plant, and that is developing from the plumule. Stem is developing from the plumule, root is developing from the radicle. And the internal structure of dicot and monocot plant we need to study now. Dicot and monocot stem first we will learn. So here, while learning about the characteristic features of dicot stem and monocot stem, dicot and monocot, first you need to tell about the epidermis. Okay, just think. Here epidermis is there, next comes the hypodermis region you can see, then after that cortex region coming, then endodermis is coming, then comes the pericycle region, then medullary race, then pith region is coming, then vascular bundles. About vascular bundles also we need to tell. So first you should remember the structure. Okay, same, the structure, this figure should be very clear. Okay, this should be learned perfectly so that you can differentiate with this figure. Leaf, root and stem should be very clear so that you can differentiate in a very easy manner. Here you can see the characteristics. From top you should start. Epidermis, right? In dicot stem, this is dicot stem and this is monocot stem, right? So here you can see, first we will tell the epidermis. In dicot stem, cells are larger and they have, here you can see the larger cells of epidermal cells. Large cells, and that is with multicell hair. Hairs you can see that is having many cells, plus multi-celled hair, right? What about monocot stem? In monocot stem, compared to the dicot stem, the monocot stem cells are smaller and they don't have hair. Smaller cells, smaller epidermal cells, and no hair. Okay, this is about the epidermis. Now second point goes to the hypodermis, hypodermal region or hypodermis. So hypodermis, that is colonchimatous. In which one? In dicot stem. In dicot stem, the hypodermis region, that is colonchimatous. Here you can see the colonchima. So this is the hypodermal region. But here, and you can see the hypodermal region, that is green color, but in the monocot stem it is sclerenchymatous and that will be non-green. Sclerenchymatous, right? Here you can see in the picture it is sclerenchymatous, right? And next comes the cortex region. And what about cortex region? Cortex region in the dicot stem, it is made up of several layers of which type of cells? Here you can see several layers of parenchymatous cells. Several layers of parenchyma cells. Okay, so parenchymatous tissue. Cortex is made up of parenchymatous tissue. And what about monocot stem? In monocot stem, you cannot differentiate like that. Here the cortex region is absent, but here you can see the parenchymatous ground tissue which is present from the hypodermis to the center of the stem. There is no separation like this. There is no endodermis. Right. So we are saying in the dicot stem, we are saying it is the portion between the epidermis and the endodermal region, right? But in the monocot stem we can't differentiate like that. There is no limiting boundary for that. So here we are saying cortex is absent. We can't differentiate like that. So it is absent, but the parenchymatous ground tissue, that is also included in ground tissue. So ground tissue is parenchymatous, that is from the hypodermis to center of the stem. Hypodermal region is from there to the center of the stem, that is called as the ground tissue. Next comes the endodermis, and here again the endodermis is absent in monocot. Such differentiations are absent in monocot, right? Here, but endodermis in dicot you can see single layer, which is a starchy sheath, and usually this is not well differentiated, but it is like a starchy sheath. Then comes the pericycle. We said about pericycle. Pericycle is made up of one or more layers of parenchymatous and sclerenchymatous layers. Which one? Pericycle. Here you can see. It may be with one or more layers of parenchyma cells. In which one? In the pericycle of dicot, but that is also pericycle region also absent. It's not like absent, that portion differentiation is not there. In which one? In monocot we have not differentiated into different regions like that. Okay, then medullary rays. Medullary rays, here you can see in the dicot stem, you can see the medullary rays which is found between the vascular bundle. See, this is one vascular bundle and this is another vascular bundle. So medullary rays you can see in between this. In which one? In dicot. Okay, but that is absent in which one? In monocot. Next comes the pith region or medulla region. So that is abundant and it is made up of parenchymatous cells. Okay, and situated in the center of the cell. Stem, and in between vascular bundles also this is present. Which one? Pith. You can see in this diagram. But like that, one separate pith region is absent. In the case of which one pith region is absent? In monocot. It's present in dicot. Then what about vascular bundles? The last point. Vascular bundle, that is in a ring shape. So in dicot stem, vascular bundle is in a ring shape. But here in the monocot, scattered vascular bundles you can see. That means not arranged in a particular pattern. See, here and there it is scattered, but here it's arranged in a ring shape in dicot, but in monocot it is in a scattered format. Which one? The stem vascular bundles. Okay. So this is how we need to differentiate the monocot stem and dicot stem. So here we are learning the characters of monocot and dicot stem, and we are able to differentiate also. Okay, so always learn with the help of these figures. The radicle of a plant embryo, and usually dicot root that is having a taproot system, and monocot root is having adventitious root system. Taproot system is present in dicots and adventitious root system is present in monocots. So here also we are going to mark like dicot root and monocot root. Then the characters. Here also we are first saying about the pericycle region. Pericycle region that is giving rise to the secondary roots and the lateral meristem. Okay, in which one? In dicot root. What about the monocot? That is giving rise to lateral roots only to lateral roots. Then about the vascular bundles. Here you can see the vascular bundles. It is diarch to hexarch. Which one? Vascular bundles in dicot root. So this is dicot root. You can see diarch to hexarch. But hexarch to polyarch in which one? That means more than 6 number of vascular bundles you can see in which one? It is in the case of monocot roots. Here you can see vascular bundles, number of vascular bundles. Then what about cambium? Cambium that at the time of secondary growth, it is developing in the dicots. So during secondary growth, cambium is developing in the dicot root, but cambium is absent in monocot root. Then next comes the pith region. Here you can see pith region. Here also it has marked as pith region. So pith region that is poorly developed or it may be absent in dicot root. In dicot root, it is poorly developed or it may be absent, and abundant and fully developed in which one? In monocot root. So pith region is poorly developed in dicot, and in monocot it is fully developed. In which one? In monocot. Pith region is fully developed. Then what about the secondary growth? Secondary growth. So what we can say about the secondary growth? Secondary growth is taking place in the dicot and there is no secondary growth in the monocots, right? And cortex and endodermis That is, narrow cortex will be present in which one? In dicot root. But cortex is very wide in monocot root. Clear? Next comes the dicot leaf and monocot leaf. This is the dicot leaf. Dicot and monocot leaf. Here also about the epidermis we need to tell. Epidermis of dicot and monocot leaf. Then epidermal cells. Then number of stomata. Then comes the guard cells. Then mesophyll cells. This is leaf, right? So mesophyll cells also. Then about the veins. Then bundle sheath. And last about protoxylem also we need to mention. Right. So here in the dicot leaf, upper epidermis that is having larger and thick walled cells, and compared to the lower epidermis, upper epidermis has larger cells and thick walled cells. Okay, in the case of dicot leaf. Then what about monocot leaf? Monocot leaf, actually the epidermis cells, they are similar on both the surfaces. They are similar. Upper epidermis and lower epidermis of the leaf, that will be similar. Then epidermis cells, that will be having sinus walls and the silica, which does not occur on the outer wall of the epidermis cells. Okay, but here in the case of monocot, the epidermal cells, they have straight walls and silica deposits in the outer wall of the epidermal cells. Okay, so epidermal cells. Next comes the number of stomata. More number of stomata will be present on the lower surface compared to the upper surface. In which one? In dicot leaf. What about the monocot leaf? Equal number of stomata occur on both the surface. Stomatal number will be equal on both the surfaces. Right. Then the guard cells. Guard cells are bean-shaped guard cells. In which one? In dicot leaf, you can see bean-shaped guard cells, and guard cells are dumbbell-shaped in monocot cell. In monocot cell, you can see dumbbell-shaped guard cells. Next comes the mesophyll cells. Mesophyll cells, that is differentiated into palisade and spongy tissue. Here you can see the palisade parenchyma and spongy parenchyma. In which one? In dicot. Palisade parenchyma and spongy parenchyma you can see in the dicot. And there is no such palisade parenchyma or normal parenchyma differentiation in the monocot leaf. You cannot see such differentiation. Okay. Next about veins. The veins will run in different directions. In which one? In dicot leaf. That means reticulate venation in dicot. But in the case of monocot, you can see the parallel venation. Then about the bundle sheath, we need to say bundle sheath is formed of colorless cells, and the extension of bundle sheath, that is parenchymatous. Bundle sheath. Here in the monocot leaf, you can see the bundle sheath. that usually they'll be having chloroplast and the extension of bundle sheath, that is sclerenchymatous. Okay, then one last point about veins we said, bundle sheath we said, then the protoxylem. Protoxylem does not differentiate in which? In dicot leaf. But the protoxylem is differentiable into two large vascular bundles. In which one? In the monocot leaf. Okay, so now we have learned in today's class, we have learned about the anatomy of which and all, anatomy of dicot and monocot plants. Generally we can say anatomy of dicot and monocot stem, anatomy of dicot and monocot root, and the anatomy or the features of dicot and monocot leaves. That's what we have learned today. And here one common topic is secondary growth that we will learn in the next class. In the next class you will be learning the secondary growth, and the you're going to solve the questions from the previous year question paper also. So in the next class we will be finishing this chapter. So now you have to differentiate about both the structure. You need to learn and explain very well, or you need to note down somewhere, and then afterwards, after learning individually, you can do the comparative study. Okay, thank you.